It was drifting silently, orbiting over the curve of the Earth, inside the controlled environment of China's Tiangong space station, where air is filtered, temperatures are stabilized, and systems hum in near-perfect synchronization. Something extraordinary was quietly growing in a petri dish. It wasn't a piece of technology. It wasn't a system malfunction. It was alive. A single-celled organism. A bacterium. And not just any bacterium. Its name? Niallia Tiangongensis. A name few have heard, yet one that may soon change our understanding of life on Earth and beyond it. Scientists conducting microbial surveillance on the Tiangong space station were running routine checks. It's standard practice in space missions. Even in the vacuum of space, astronauts aren't alone. Microbes from their bodies, supplies, and equipment hitch a ride, adapting and evolving in the microgravity environment. Most of these microbes are familiar. They're the usual suspects: Staphylococcus, Bacillus, even Escherichia coli. But this time, something was different. A culture sample revealed a strange colony. It didn't quite behave like anything in their microbial databases. Under the microscope, it showed structures consistent with the genus Niallia, a relatively obscure genus within the family Bacilliaceae. But genetic sequencing told a different story. This was a new species. They named it Niallia Tiangongensis, after the space station where it was discovered. But what makes this discovery so remarkable isn't just the novelty of finding a new bacterium. It's the way this microorganism has adapted, survived, and possibly evolved in the uniquely hostile environment of space. Let's pause for a second and think about what that means. Space is one of the most extreme environments life can face. There's the issue of microgravity, which affects everything from fluid dynamics to gene expression. Then there's ionizing radiation, more intense than anything encountered on Earth's surface. Add to that limited nutrients, fluctuating temperatures, and the psychological stress of confinement, and you have an environment that, in theory, should be lethal to most life forms. Yet Niallia Tiangongensis thrives. Researchers began to probe its secrets. They found that this bacterium possessed unique stress response genes, some never seen before in terrestrial relatives. Certain enzymes it produces appear to protect its DNA from radiation-induced damage. Others seem to enhance its metabolic efficiency, allowing it to survive on minimal resources. There are also indications that its cell wall has become thicker or more structured, possibly an adaptation to withstand the increased osmotic stress and lack of gravity. This kind of adaptation isn't just biological curiosity. It's evolutionary acceleration, happening not over millions of years, but in the span of a single mission. And that leads us to some big questions. Was Niallia Tiangongensis always like this? Or did it evolve these traits in response to its time aboard Tiangong? To answer that, scientists compared it to its closest Earth-bound cousin, likely Niallia circulans. While the two share significant genetic overlap, the space-derived strain exhibited over 100 unique genes, many linked to survival under extreme conditions. Some of these may have been acquired through horizontal gene transfer. Others may have resulted from mutational bursts triggered by cosmic rays. In essence, Niallia Tiangongensis is a case study in rapid stress-induced evolution. And it's not alone. We've seen this pattern before. On the International Space Station, scientists have previously identified mutations in Aspergillus, Bacillus, and even E. coli that enhance survival. Some developed biofilms more easily. Others became more antibiotic resistant. It turns out, space isn't just a destination. It's a crucible, a forge for microbial evolution. Now let's ask the bigger question. Why does this matter? In the coming decades, humanity plans to venture deeper into space. Moon bases, Martian outposts, maybe even asteroid mining. All these missions involve humans living in enclosed habitats for months or years at a time. Wherever humans go, microbes follow. That's not a hypothesis, it's an inevitability. If some of those microbes begin to evolve in space, as Niallia Tiangongensis did, what happens next? Will they become more virulent? More resistant to antibiotics? Or could they become allies? That's the double-edged sword of microbial adaptation. On one hand, it raises biosafety concerns. If bacteria evolve faster in space, they might outpace our ability to control them. Imagine a mutated strain that thrives in closed-loop habitats, forms persistent biofilms on life support systems, or becomes pathogenic to astronauts whose immune systems are weakened by zero gravity. But there's another possibility. What if we use these adaptations? What if space-evolved microbes become tools for biotechnology? Niallia Tiangonggensis, for example, may hold secrets to new radiation-resistant enzymes. These could be used in cancer therapy, waste processing, or even protecting crops in extreme environments. Its metabolic adaptations could help engineer microbes for life support systems, recycling waste, fixing nitrogen, or generating oxygen from limited resources. Think about a Mars colony, dependent on bioreactors seeded with space-hardened microbes. Think about industrial-scale microbial mining operations in microgravity, where modified strains extract metals from asteroid dust. It's not science fiction. It's synthetic biology in orbit. Back on Earth, scientists are already analyzing the full genome of Niallia Tiangonggensis. They're comparing its proteome and transcriptome to terrestrial strains. And they're beginning to understand just how plastic, how resilient microbial life really is. But let's go even further. What if microbes don't just survive space, they thrive there? Could bacteria like Niallia Tiangonggensis be the vanguard of life's expansion into the cosmos? Panspermia, the theory that life can travel between planets on asteroids, has long been debated. Microbial survival in space strengthens the plausibility. If Niallia Tiangonggensis can persist in orbit, what's to stop other organisms from hitching a ride on meteorites, enduring eons in suspended animation, and sparking life somewhere else? This changes how we view the origin of life and where life might exist beyond Earth. Imagine an exoplanet, harsh and barren, but seeded millennia ago by microbial spores drifting through the void. If they could adapt, evolve, and diversify, might alien ecosystems begin with bacteria not unlike Niallia? This discovery, made quietly aboard the Tiangong station, reminds us that exploration is not just about rockets and rovers. It's also about petri dishes, about what grows when you combine biology and the vacuum of space, about how life doesn't just cling to existence, it transforms. Niallia Tiangonggensis is not just a bacterium. It is a symbol, a message in microbial form. It tells us that life is more adaptable than we imagined, that space isn't sterile, and that wherever humans go, the tiny travelers we bring along, those unseen stowaways, may be writing their own evolutionary stories. Stories that begin in orbit and might just end on another world.